Hey, what's going on, guys? Welcome back to another stream on the Math Studies LC channel. Um, Today we have a lot of work in for us because we're a bit behind. I won't lie. But as the title of the stream says, I'm going to study the whole Leaving Cert course in one day to show that it is possible for biology. I have all the flashcards made, everything ready to go. I've already done half of it, but I need to go over it again because I don't think I did it in the best way. And there might be some diagrams I might forgotten how to draw. So yeah, we'll do all that again, and then we just have all these chapters left here. So we're gonna get straight into it. We're gonna be starting off with a chapter called fungi, and yeah, so we're just gonna be grinding today. Just no distractions for the rest. There's only 24 days left. I'm feeling a bit screwed, I won't lie, but hopefully we can hopefully the hard work I did over the years will pay off if I can just get one last piece of revision in before we get into this. So before we get started, let me just do this. And that works great as well. So that's all good. That's all good. Actually, I won't move it just yet, but I think we'll start it. Alright, guys, best of luck. Actually, you know what? I'll start it once I get everything set up, because right now everything is nowhere near set up. Please may have not logged me out. I will very be sad if it did. Hopefully, yeah. No, we're good. We're chilling. Okay. And this is back to who I am as a person, bro. Like there was times, bro. Most of the time I'm very disciplined, but just sometimes when my headspace, I'm not in the right headspace, and I'm just losing my discipline. But anyways, yeah, I'm ready now. Alright, let's go. 3, 2, 1, let's go. Wait. I know I can't lie. Okay. So the first question I was asked was, okay, what are the features of a rhizopus? Now I haven't studied this chapter at all, but my idea when it comes to these chapters, I don't have any, but I'm not prepared for this at all, bro. For this stream, but I think that should work for us. So. I'm back. So what are the features of a rhizopus? Very simply, if you come here, so hyphae are the tube-like structures which have no cross walls to aseptate. So hyphae are the tube-like structures, and they're multinucleate, with each nucleus being a haploid. So haploid meaning in a human meaning n equals 23, so it's not a homologous pair. So it only contains one of every type of chromosome. And a mass of hyphae is called a mycelium. So hyphae are like these tube-like structures which have no cross walls. Tube-like structures which have no cross walls, that is septate, and they're multinuclear with each nucleus being a haploid, and a group of a mass of hyphae are called mycelium. Okay, what are the economic benefits of fungi? So, for example, yeast. You know, we looked in the respiration chapter. Used to make ethanol and CO2. So bread is CO2 is going to make the bread rise. Ethanol is going to be used in alcohol. So used to make bread, alcohol, and then like mushrooms are grown for food. Okay, no problem. Okay, explain two types of saprophytes. So what I know already about saprophytes, so saprophytic bacteria, is that they get their food from a dead host, right? Their role in nature is to recycle minerals, and example was bacteria of decay. Now, in terms of fungi, is obligate parasites and facultative parasites. So we had viruses are obligate parasites because they require a host cell. So they're obligate parasites, right? So can only get its food from a obligate parasites can only get its food from a live host. Remember, obligate anaerobes and facultative anaerobes, which means they can respire with both oxygen and non-oxygen. But basically, obligate parasites can only get its food from a live host. They can only get their food from a live host. For example, mildews. But facultative parasites can get food from dead or alive hosts. So obligate only alive, but facultative but alive and dead. Okay. Explain the 5 sets of sexual reproduction in a rhizopus. Okay, so let's look at this. So we talked about sexual reproduction in uh, in a rhizopus. So how does that work? First of all, these are hyphae. Remember the hyphae were like these tubes with no cross section. They contain nuclei, with each nucleus being a haploid, and a mass of hyphae was called a mycelium. So we talked about these hyphae. The hyphae, right? One's plus and one's minus. And then there's nuclei inside of here. Ah, yeah, that's what they are. So hyphae from a plus and a minus strain go close together. So this is a plus strain and a minus strain. So the hyphae, so these are hyphae. So hyphae from a plus and minus strain go close together, and swellings from both form on both strains and touch each other. So hyphae from different strains. So hyphae from a plus and a minus strain grow close together, and they swell and they come together and they touch each other. Swellings form on both strains and they touch each other. So what we're saying is hyphae from both a plus and a minus strain come close together, and swellings form on both hyphae as they touch each other and touch each other. Yeah. So this is what we call nuclei. This is what we call like the hyphae. Hyphae are these little tubes, and then the swellings are these. So they don't usually have that thing sticking out. Okay, fair enough. Then we have nuclei. Step 2, which was nuclei or gametes move into each swelling, forming nuclei or gametes move into each swelling, forming prometangia. Crosswalls form to make a gametangia, progametangia, and gametangia. So nuclei move into swellings to form prometagangia, prometangia, and then crosswalls form So the nuclei are going to move into the swellings to form a prometangia, but that crosswalls form to form a gametangia. Alright, so they're still the same kind of like tubes, right? You still have the swellings, the hyphae. So these are hyphae. The hyphae, a crosswalk forms to gametangia, and then this is prometangia. Prometangia. So prometangia. Because all the nuclei move there. That's gametangia, prometangia. So let's go over that. So the hyphae, right? from a plus and a minus strain grow close together. Swellings form at the end. Swellings form on the hyphae, and they join together. Swelling forms on both strains, and they join together. Then what happens is the nuclei move into the swellings to form a prometangia, and then a cross walls forms, which is a gametangia. Alright. Okay. Then the walls between the gametangia dissolve, and many fertilizations occur to produce diploid zygote nuclei. Oh, because the haploid and the haploid are coming. I see that. So basically, so there's no cross walls anymore. There's no gametangia anymore. So. There's no gametangia anymore. So basically, the walls between the gametangia, the walls between the gametangia, but this is a gametangia. Cross walls form to make a gametangia. Now the gametangia is the walls between the gametangia. Now the walls between the gametangia dissolve, and many fertilization occur to produce a diploid zygote nuclei. A tuft-walled black zygospore forms around the nuclei. Okay, so basically, two hyphae from plus and a minus strain come close together. Swellings form on both of the strains, and they join together. Then the nuclei move into the swellings to form a prometangia, and a cross walls form to make a gametangia. Now, the walls between the gametangia then dissolve, and many fertilizations occur to form a diploid zygote nuclei. Now, tough-walled zygospore forms around the nuclei. A tough-walled black zygospore forms around the nuclei. So now, not only is it just like something like that, right? Well, that's okay, so a tough-walled black zygospore surrounds the nuclei. Forms around the nuclei. Zygospore can remain dormant for a month, so it's kind of like an endospore for like during binary fission for bacteria. So zygospore. Dispersal and surviving dry conditions. Then we have germination. So zygospore germinates by meiosis. So now this is going to germinate by meiosis. So we have something like that, right? So it germinates by meiosis. A haploid hypha. So what was this hyphae? Hyphae. So a haploid hypha grows out of the zygospore and produces these are hyphae. So hypha is a 1. So hypha, a haploid hypha grows out of the zygospore and produces a sporangium at the tip. So sporangium. So a haploid hypha forms out of the zygospore and produces a sporangium at the tip, and sporangium releases many spores. So basically, we have, so in a nutshell, guys, the way that fungi kind of reproduce sexually is, from my understanding, let's say you have two hyphae, right, from a plus and a minus strain, and they come close together. Now swellings are going to form on both of the hyphae, both of the strains, and they join together, right? Now the nuclei move into those swellings to form a metatangia, I think it's called. Progametangia. So a progametangia, and then cross walls form and make a gametangia. Now the walls between the gametangia dissolve, and many fertilizations occur to form a diploid zygote nucleus. Now, a zygospore, a tuft-walled black zygospore surrounds each nuclei, and this zygospore can help the nuclei survive, and the zygospore can remain dormant for hundreds of years until suitable conditions. So this allows for dispersal. This allows for survival of harsh conditions. Now, what happens is a haploid hypha forms out of the zygospore, which contains a sporangium at the end, right? So as the the zygospore germinates by meiosis. A haploid hypha grows out of zygospore and produces sporangium at the tip, and sporangium releases many spores. Okay. What are the features of the rhizopus? So, first of all, hyphae are tube-like structures with no cross walls, which have no cross walls, so they're separate. They're tube-like structures that have no cross walls. They contain many nuclei, which happen to be haploid. And these nuclei are haploid, meaning that many hyphae, like a group of hyphae, is called a mycelium. A mass of hyphae is called. What are economic benefits of fungi? So fungi are used to make bread, alcohol, and fungi, yeah, they're like mushrooms are eaten as well. So mushrooms are grown for food, yeah. What two types of sacrifice in relation to fungi? So we have obligate parasites and facultative parasites. Now, obligate parasites and facultative parasites. So obligate parasites can only get their food from a live host, i.e., mildews, whereas facultative parasites can get their food from both a live and a dead host. Explain the five steps of sexual reproduction. So first of all, well, I'm gonna draw this out. So you have two kind of strains, right? Two strains, two hyphae from a positive and a negative strain come close together, right? So they come close together, right? And we have just nuclei and everything, right? And the swellings form on both of the strains and they join together, right? Then the nuclei move in to the swellings to form a progametangia, and then cross walls form and make a gametangia. So gametangia will be like here, all the nuclei here. Now the walls between the gametangia dissolve, right? So the walls between the so if the walls between the gametangia are going to dissolve, right? And many fertilizations take place to form diploid zygote nuclei, right? Now, a tuft-walled black zygospore forms around the nuclei, and the zygospore can remain dormant for 100 years until conditions are suitable. Now, we know that the zygospore is going to germinate by meiosis, and a haploid hypha is going to grow out of the zygospore with a sporangium at the end, and the sporangium then releases spores as well, yeah, I think releases many spores. So it's still plus minus till step 2, then the plus minus is kind of just gone then. Okay, what are the features of a rhizopus? So they have hyphae, right? So hyphae, first of all, I just want to see what is a rhizopus. A genus of common fast-growing filament, like they're kind of like thin. So they contain hyphae. Now the hyphae. So rhizopus have hyphae, which are tube-like structures with no cross walls. Inside a hyphae, they have nuclei, which are haploid, and a mass of hyphae is called a mycelium. Give two examples of an edible fungi, like mushrooms, field mushrooms, and truffle. Okay. Three features of fungi. So fungi are eukaryotic, which means they have a nucleus, as we said, nuclei, and membrane enclosed